Okay, good morning to all of you. So we'll continue uh, with the chapter on origin of life. So I guess I don't remember I have sent you the PPT or not, but today I'll do. First, let me share the PPT. Okay, I hope you are able to see the PPT.
Yes, sir.
Okay, good. So, yes, not visible. There's some problem with the slideshow. I don't know. When I make it, now it is visible. Hmm. Okay, let me. Okay. Is it visible now? Okay, good. So we sort of figured out that almost 3.5 billion years ago, we have evidence of some evidence of living organisms, and because we have these stromatolites that are definitely organic, organogenic, and today they are found in shallow marine environments. So, sin sedimentary origin of life has always been a possibility in the sense that uh, these stromatolites, these mats, layered structures uh, formed due to bacterial activities. So you have you have a platform over which these bacteria grow, the cyanobacteria grow, and they sort of uh, form a layer, and then this layer gets covered by another a sedimentary layer because of continuous sedimentation. So again, you have these bacterial growth now starts afresh on another layer. So that's why you get layer by layer of these organo sedimentary structures which are called stromatolites. So this was one of the most uh, compelling evidence. This it is today that uh, this evidence, their evidence is there. We know that they are organically generated. Now, when we look at what could have been the conditions, whether the atmosphere was anaerobic to begin with, and then it became aerobic. There are various hypotheses where oxygen gets enriched over a period of time, and that allowed some of the organisms to diversify. That's what we call the eukaryotes. Mostly eukaryotes, they have this propensity. So uh, these layered microbes uh, use photosynthesis to form layer by and of course requires sunlight, constant supply of sunlight. Now, uh, beside these, there are cherty layers where people have been able to find fossils, strands uh, of organic uh, of organic origin, because chert is basically silica, so that produces that sort of preserves, uh, that has got the capacity to preserve good fossils, even microfossils, microorganisms. So when we talk about this 3 point some billion years ago, the scenario, we have these what you call banded layers. Most of them now, they are all metamorphic. A lot of metamorphism has occurred, and there is hardly evidence of early crustal rock. So these get churned and rechurned over billions of years. Now, early, if you look at the isotope values, low delta C13 values, fractionation suggest life biological process prefer. So there was a low concentration of isotopic values. It means life was just beginning to evolve. So when did life begin? We still are not very sure. We dwelled on this in last couple of lectures, that whether it's an extraterrestrial or uh, within it evolved within the earth, and there were various hypotheses. So we sort of explored all sorts of hypotheses, um, extraterrestrial and thermophilic and spontaneous, and all sorts of hypotheses were discussed in last lectures. So one of the most widely accepted hypothesis is RNA world, which we uh, also briefly touched upon, that people a lot of people believe that it was RNA that was prevalent to begin with, and that uh, started replicating, self-replicating RNA would ultimately lead to uh, formation of of a living cell, living body. Now we all know that what RNA does, and how what is the difference between RNA and DNA. It is simpler, and uh, DNA. We all know this, and so we just okay. I think we looked at this, the early hypothesis of how uh, people try to do experiment and try to produce or recreate a cell, a living organism uh, in the lab that was not successful. So then we uh, dwelled upon what kind of uh, what were the possibility of getting the organic molecules, whether from extraterrestrial like carbonaceous chondrites. Uh, they do possess some organic stuff, but then again the 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 questions still remains, when did self-replicating molecules began to form. So you have DNA, we know it is double stranded. RNA is obvious candidate. So these were some of the early search for the RNA world. There it is. So now these are all these various possibilities were explained, that how uh, such conditions would have formed in the early water bodies. So again, it remains hypothesis that RNA brings about the changes, brings about replication, and then a point comes when the information is stored inside a nucleus. So this transformation, this change has been explained by these very uh, hypotheses. Now, uh, all right. So again, how before a cell is formed, what would be, what are the stages through which you get a nascent, a very preliminary stage, and then uh, the final stage. So you have to have a stage when cells started dividing, splitting. Anyway, these are all sorts of hypotheses, and we also know that uh, molecules have there are left-handed, right-handed molecules, and this very diagram summarizes that how this would have happened. So first, a synthesis organic precursor molecule would develop, where you have these self-replicating RNA. They would separate and put their information elsewhere, and then these get enclosed by a membrane, which would ultimately become a cell. Uh, so we all know that uh, in primitive cell, there would be RNA genome at the center, followed by or covered by uh, the organelles, and finally it would give rise to DNA genome, which would get preserve all the information. So we also discussed that there is a possibility of organic stuff or life forming elsewhere and getting transported to uh, the earth through some comets, meteorites, or asteroids, and that possibility was sort of got lot of popularity because of finds in the 70s and 80s, which suggest that there are these strands. They look very similar to early bacterial sort of organisms. Uh, so there is that's Martian so-called meteorite containing fossil type of thing under scanning electron microscope, which sort of look very similar to present day micro organisms. Like we have these living cyanobacteria on your left hand side, and microfossils found in chondrites, carbonaceous meteorites. So still the jury is open, and we still not very sure how began. So uh, what kind of early atmosphere was? We discussed this a little bit. That to begin with, we had all these anaerobic bacteria. So because there were hardly any oxygen in the atmosphere, so most of the life had to be anaerobic. So there might be chemoheterotrophs. So nutrients on obtain nutrients from organic material, nutrients from inorganic material. So as we all know, if we look at the living world today, uh, it is it is divided into three major groups. That is, you have the bacteria, archaeobacteria, and the eukaryota. So eukaryota is like we all belong to that group, and you have a huge bacteria and archaeobacteria. These are two big groups. Now these were very prevalent in the Precambrian time period, which would be somewhere around 3.5 billion years ago. Now there are uh, other evidences beside fossils, like uh, like when we look at the earliest fossil evidence that we just talked about. Like one, we have these early stromatolites, some 3.5 billion years, followed by uh, yeah, stuff in Australia, where you have these bacterial evidence or evidence of uh, microorganic molecules in Cherty areas. Cherty, some 2.5 billion years, 2.2 billion years, and then again we have later on, of course, we have a lot of evidences, but the hard evidence is difficult to come by. Whatever we have uh, these uh, like the Gunflint chart, the from Canada, and these stromatolites which are spread all over. So there are, of course, biomarkers that are there. Biomarkers are nothing but uh, evidence of organic remains. So they are molecules, organic molecules. uh, Now they these biomarkers tell us that eukaryotes were there while while the world was dominated by the bacteria. So by proxy we can find out, you know, you just analyze the rock, the sediments for these biomolecules. Their presence uh, gives us some idea about existence of eukaryotes. So coming back to the type of atmosphere, obviously it was anaerobic to begin with, and so you have the most important metabolic processes evolved gradually with photosynthesis to begin with. Organisms that live close to the ocean surface, of course, they use sunlight to produce this photosynthetic pathway, and then uh, that turn into energy. So these evidences we already have. Now we we can see that water uh, was there, of course, whether it was shallow marine or shallow freshwater, not we are not very sure, but the cyanobacteria appeared around that time period. Now gradually the atmosphere must have got enriched in oxygen, because whatever oxygen was produced by these early photosynthesizing organisms must have got absorbed by the surrounding before it could be passed on to the atmosphere. Moreover, atmosphere was full of methane those days, and that is why the in spite of the less solar insolation, that is, it was almost one fourth of the kind of sunlight we get today. It was the one fourth of the intensity if we are talking about Archean time period. So uh, that would lead to glaciation. But then, because the atmosphere was full of methane, that provided methane. We all know that is a greenhouse gas. So it sort of retain the warmth uh, all around. But then there was no way oxygen could sort of find its place, even if whatever oxygen comes out due to from volcanoes, that would get consumed by the rock, by the surrounding minerals, because oxygen is very highly reactive. So there is this hypothesis that hydrogen lost to uh, was lost from the atmosphere somehow slowly, and that allowed oxygen to fill in. So it was not directly connected to uh, photosynthesis or photosynthetic bacteria. So there were various processes that slowly and gradually enriched the atmosphere. First, the surrounding ocean, and then the atmosphere. And there's the best evidence is formation of banded BGCs, banded hematite, banded iron BIFs, banded iron formations. So where you find layers of hematite magnetite rocks, and then covered by silicates, by cherty layers, and then again you have these banded hematitic layers. So uh, today, if you look at the scenario in the ocean, it is enriched in oxygen. So the moment iron comes out from any volcano or from any fissure, it gets uh, 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 it reacts with the oxygen, and iron forms then and there. But in the ancient days, when these banded hematite formations were forming some 2 billion years ago, they could the oxygen the iron would come out uh, and would spread, and then due to planktonic bloom, uh, which of course produced oxygen, these bands would form after reaction with by. So these are basically iron oxides. So you get a layer of iron oxide. Again, that process goes on and on and on. So that is how in the early ocean these banded hematite formations were formed. And then that stopped after that. After 2 billion years, you won't find them. So the record, when you look at the record in the geological past, you will find most of the banded hematite formations, banded iron formations are confined to these early Proterozoic time period. So oxygenation, you can imagine that these were the early oxygenation events that uh, sort of not only helped the surrounding ecology atmosphere, but also provided a stronghold, a foothold for the life to come on the land. So first on the land, algae and uh, other plants would come out, and that would be followed by animals. Anyway, we'll come to that later onward. But still, there are these various hypotheses how they discuss that how oxygen gets reacted very fast, because it is very reactive molecule. So uh, you have to have first rocks get saturated, then only oxygen can be accumulated in the atmosphere. And once oxygen uh, is there, then you have the protective ozone layer. So it's a process that went on, and it was a slow kind of process, but surely uh, it took billions of years. Just look at uh, 2 to 3 billion years ago, there were hardly any oxygen in the atmosphere, or even less than 1% of the current oxygen. You can see what kind of uh, impoverished atmosphere it was during those days. So now it's only 200 million years ago that we come to know that the kind of concentration we have today, the atmosphere acquired that sort of concentration just 200 million years ago. Prior to that, it was oxygen impoverished. So now, to begin with, a rise of oxygen would have created a crisis for life. So that is very important because life to begin with was comfortable in oxygen which was devoid of oxygen. So they were prepared. They were more. They were the life was better suited to other conditions than oxygen, because oxygen reacts very fast and organic material bonds reacts with bonds of organic material. So surviving species avoided oxygen. So gradually many anaerobic microbes found in such locales today. So if you look at very deep seated areas, sulfurous or volcanic vents, there most of the conditions are anaerobic. Now we come to eukaryotes. So we just discussed a little bit about uh, what were the early prokaryotes. We have these bacteria, archaea and bacteria, and then eukaryotes that contains a nucleus where you have these uh, chromosomes, and then you have DNA. So it's a more complex system than the simple system of prokaryotes. Now, fossil evidence dates back to 2.1 billion years, dates to when oxygen was rising in atmosphere, and DNA evidence suggests that prokaryotes and eukaryotes separated from common ancestor earlier. Now, this is again somehow uh, it has been proven that of course, we do not have hard evidence of eukaryote existing 3.5 or 3 billion years ago. But then we have some biomarkers that suggest there is a possibility eukaryotes can be traced back to almost 2.7 billion years ago. So, oxygen played a key role in eukaryote evolution. Cells can produce energy more efficiently using aerobic metabolism than anaerobic. This we all know. Adaptations of aerobic organisms could develop adaptation that required more energy that would be available for anaerobic organisms. So, oxygen, of course, revolutionized. Then came the Cambrian explosion. So, before that, we need to understand something more, that is, whether eukaryotes evolved from uh, prokaryotes, from bacteria, or there was a common ancestor, whether they branched from a common ancestor. This is a, a very interesting aspect, because here people are trying to figure out how eukaryotes and prokaryotes are related, interrelated, and where did the split happens. So uh, I'll just see if I can show you the. There was a figure here. Yes. So common ancestor. I hope you all can see this. So you have bacteria on one hand, archaea on the other, and then eukaryota separated. Now, whether eukaryotes were somehow related to archaea, it is still not very convincingly evidence, because now we know that there are biomarkers that tells us that there is a possibility that eukaryotes were also there while bacteria and archaea was around some 3 billion years ago. So what I'll do is switch this PPT with the PDF from the Benton's book. I'll go to the Benton's book where I had told you to go through that chapter. So I'll just tell you to stop presenting this. So some points from the Benton's book we can look at. Let me see if I can present Benton's book. All right, so there it is. Hope you can see Benton's book now, highlighted areas. Okay, good. So I'll just, this chapter is very interesting. You should go through all the. There you go. There it is. That's the universal tree of life. So uh, now you see, you have these eukaryotes, where animals, fungi. See, animals and fungi are very close by. So it is they are more animalia is more related to fungi than to plants. And then you have all sorts of other organisms, that multicellular organisms. And archaea is a huge group, and bacteria, of course. So I'll see. I just highlighted some of the points I wanted to elaborate upon, but you can, because you need to go through each and every point here. So just universal common ancestor. Okay, this is about RNA world. Evidence of early life. Now look at this diagram, this very 8.3 time scale. So that's why I was talking about. Look at the Hadean time period, origin of Earth. Then you have the oldest evidence of life, the prokaryotes. So you have these prokaryotes, all the way they go back to almost 3.6, 3.7 billion years. Then you have these fig tree cherts, followed by gunflint chert in the Proterozoic time period. There is oxygenation of atmosphere by this time. And then, of course, eukaryotes come in, and multicellulars and metazoans and vertebrates and mammals and all. So Uh, Let me see. Evidence of life. We have kind of touched all these. Oldest rock, then uh, isotopes. We did touch about that. Even if we do not have organic organisms, but isotopes can tell us whether organisms were there or not, because mass spectrometry comes very handy here. Then great oxygenation event that we just talked about, that around 2.5 billion years ago to almost beginning of, that is Proterozoic, is uh, starts from 2.5 billion years ago. So you have these first anaerobic bacteria, and and you have early evidence of Precambrian uh, fossils. Right, where did early banded iron formation I just talked about? Let me see, where are those? Then you have the universal tree of life. So you should go through how these tree of life has been created. So to begin with, people would just separate things into animals, vegetables, or minerals, and then came, well, where would you put microbes then? So there was further separation into four groups, and then microbes were divided into further kingdoms. So uh, four kingdoms, and then you have uh, five kingdom picture with fungi coming in as a separate group. And of course now we rely more on molecular phylogenetics, which sort of uh, talks about three main groups: archaea, that is archaeobacteria, and bacteria, that is eubacteria, and eukarya, that is eukaryotes. uh, Making from this UTL, universal tree of life, is what uh, early molecular biologists have been trying to figure out, that how this UTL was produced. Now there is not a straightforward experiment. There has been some issues, some problems, because there has been some jumping genes. So there is a horizontal gene transfer. So there is a possibility of jumping genes. There it is, where some genes can get transferred, and that can create problems while formulating these molecular trees. You know how molecular trees are formed. So there are various ways, looking at mitochondrial DNA, or there are other methods to look at the similarity and differences among present day organisms. So the most accepted hypothesis is this, where you have these all these divisions, and we know that how eukaryotes are different from other bacteria and archaea, because these these organisms, they have chloroplast in them. They have these flagella, uh, and then you have the cell wall. Then you have organelles inside. So so that is how they are different from the very simple forms of prokaryotes. Uh, this we talked about what Precambrian prokaryotes look like, and we have these stromatolites and cyanobacteria, and so oldest currently accepted fossils other than stromatolites are the dated to 3.2 billion years. So stromatolites, they come almost, they have been found almost 3.43 billion years ago from rocks that old. And next would be 3.2 billion years, where you have these sulfide deposits produce an environment like modern deep water. So there you have bacterial thermophilic bacteria. So that is uh, the most one of the most important finds. Then comes uh, there is a gap, of course, and then comes the South African uh, cyanobacteria deposits, fossil cyanobacteria from South Africa, dated to almost 2.5 billion years, and then followed by gunflint chert, which are 1.9 billion years old from Canada. There are various form, filamentous, umbrella-like forms, and then I just talked about biomarkers. Even if you do not have the oldest fossils with you, but you have the the proxy. So biomarkers, organic chemical indicators of life in general. So even if you do not have the hard parts preserved, there is every possibility you may get the molecules that got released from the organisms. So they are very much used in all sorts of industries, all sorts of techniques in biology, in biotechnology. But these are also used in finding out early biomarkers of early organisms like fossils. So even sometime in very younger rocks, if you do not have, for instance, you do not find a particular fossil, but if you find a particular biomarker, that would sort of uh, give a proxy that, well, this particular biomarker means that uh, these sort of plants were there. So uh, you have biomarkers going back almost 2.7 billion years, where two types of uh, first cyanobacteria was there, and uh, these they identified methane methylophanes. So two methylophanes, which is sort of a breakdown product of cyanobacteria. So such uh, the also they found some steranes, which are sedimentary molecules derived from sterols. So large ring sterols are synthesized only by eukaryotes. So they also found not only cyanobacteria, they also found sterols. So there was some evidence of presence of eukaryotes all the way back to 2.7 billion years. So all these division between the main groups, archaea, bacteria, and eukaryotes, whether it took place later or very early, some 3 billion years ago, it's not settled yet. So I would suggest you go through this chapter carefully, and then of course, I will give you the slides, the two slides I've been teaching from, and see if you cannot understand anything. We can discuss those. I think it's once you go through this chapter and the slides, I think it should be good enough for you to gather the main points of the topic, that is origin of early life. So we stop here today, and then we will move on to the next topic. So that is all about origin of life and its early diversification, what kind of experiments early people did to figure out how life originated, what were the main hypotheses out of these, what RNA world was, and how do we know that uh, whether what kind of atmospheric conditions were there when early life uh, evolved. All right, so I'll take the attendance. All right, thank you. See you next time. Bye.